Hello, I'm Chris and welcome to my presentation for my final project of CS50 Python. My program is called Python Codon Usage Comparison Tool, or PyCUCT. So what is it for? Imagine you want to recombinantly produce an enzyme from some organism in some other organism. Now you have to make some decisions of how to go about it. One of those decisions is, do you want to change the DNA of your gene of interest? And PyCUCT can help you with that decision. So why would you want to change the DNA of your gene of interest? Well, there are 64 codons encoding 20 amino acids and a stop codon, which means that multiple codons can encode the same amino acid. And another thing that's true is that some codons are used more frequently than others. And an effect of that is that protein synthesis actually slows down if a rare codon is used in the gene versus a more common codon. And so now you have to make a choice. So you can change all the codons in your gene of interest, changing rare codons to common codons to speed up protein synthesis and therefore hopefully get higher protein expression. And this is called codon optimization. However, the downside of that is that actually in your native organism, there might be rare codons being used kind of on purpose in specific parts to slow down protein synthesis to give the protein enough time to properly fold. And so then you might be tempted to use the wild type sequence to sort of maintain these strategic slowdowns. However, codon usage varies between organisms, and sort of this strategic slowdowns in the native organism might not be mirrored in your expression organism. Luckily, codon usage tables exist and can be found in databases such as this database here, and they are kind of formatted like this, and you can just copy paste the database from the website into a text document like this, and you can actually provide multiple codon usage tables for multiple organisms if you so desire. And then you can provide this text file to PyCUCT, and it will compare the codon frequencies of the organisms that you're interested in against 3 common expression organisms: E. coli, Baker's yeast, and Pichia. And it will produce graphs for each of those comparisons, where on the top it compares the amino acid frequency, and at the bottom it compares the codon frequency. And it uses this definition of excess to compare the differences in frequencies, whereby excess of -100 means that something is infinitely less common in your organism versus the expression organism, and an excess of 100 means it is infinitely more common in your organism versus the expression organism. And so you can see here, when we compare Chrombacterium versus E. coli, that the amino acids are fairly similar, as you might expect. There may be some difference for some of them, but everything fairly minor. And then for the codon usage, you can see that it is actually quite different. And so in particular, some of the codons are actually way rarer in Chrombacterium than they are in E. coli. And so if you wanted to use those codons if those codons are used by Chrombacterium to slow down protein synthesis, this is unlikely to be mirrored in E. coli. And on the other hand, if we compare two similar organisms, Saccharomyces pastorianus versus Baker's yeast, which are closely related, as you might expect, the codon usage is fairly similar. And so I hope that this tool can prove useful to you.